Hello students, till now we studied about different types of tissues, that is meristematic tissues and permanent tissues, and then we studied about types of tissue systems, that is epidermal tissue system, ground tissue system and vascular tissue system, and then we started with the anatomy of plant organs, that is dicot stem, dicot root, dicot stem, dicot root and dicot and monocot leaves. Now let's start with secondary growth. This is the last topic of this chapter, that is secondary growth. Okay. Secondary growth. So what is secondary growth? Secondary growth is the formation of secondary permanent tissues. Secondary permanent tissues is called as secondary growth. So formation of secondary permanent tissues by the activity of lateral meristems is called as secondary growth. Secondary permanent tissues. Tissues by the activity of lateral meristems is called as secondary growth. Right? So due to the formation of secondary permanent tissues, what happens in the plant organs? These plant organs increase in girth or width or diameter, right? So secondary growth results in increase in girth or width or diameter. Width or diameter of plant organs, that is stem and root. Anyway, in leaves, secondary growth is absent. Diameter of plant organs, that is stem and root. Right? Okay, so the secondary growth occurs in dicots and gymnosperms. Secondary growth is absent in monocots. It occurs in dicots and gymnosperms. Here we are going to deal with the secondary growth of dicots, right? So in dicots also the secondary growth occurs in stem and even in root. That means among all plant organs of dicots and monocots, only dicot stem and dicot root exhibit secondary growth. Both the leaves, monocot and dicot leaf, do not exhibit secondary growth. Monocot stem and monocot root also does not exhibit secondary growth, right? So here we are going to study about the secondary growth of dicot stem and secondary growth of dicot root. Okay, whether it is dicot stem or dicot root, secondary growth occurs inside the stele, that is center part of the plant organ, and outside the stele, that is towards the peripheral side of the plant organ, right? So it occurs whether it is stem or root. Secondary growth occurs inside the stele. So secondary growth that occurs inside the stele is called as stelar growth. Stelar secondary growth. And the secondary growth which occurs outside the stele is called as extrastelar secondary growth. Extrastelar secondary growth. In case of root also, stelar secondary growth and extrastelar secondary growth occurs, right? So this stelar secondary growth occurs by the activity of vascular cambium. Vascular cambium, whereas extrastelar secondary growth occurs by the activity of cork cambium. Secondary meristem, that is cork cambium. So both are lateral meristems. Vascular cambium, which is formed inside the stele, is also a lateral meristem, and cork cambium, which is formed outside the stele, is also a lateral meristem. Okay, let's study the secondary growth of stem first. After completion of the secondary growth in stem, dicot stem, let's start with the secondary growth in root, dicot root. Okay, let's study all the steps of the secondary growth in stem. Secondary growth of stem. Stem. Okay. Now let's study the secondary growth of stem stepwise. So it involves different steps. Secondary growth in stem involves different steps. First step is formation of vascular cambium. Formation of vascular cambium. How this vascular cambium is formed. First, let's start with stelar growth. Now we are studying about stelar growth, step by step. After completion of the stellar growth, let's start with extrastellar growth of stem. Right. In stellar growth, let's study this stellar secondary growth step by step. That is formation of vascular cambium, and then activity of vascular cambium, how this vascular cambium shows its activity in increasing the girth or width or diameter of the plant organ, that is stem. Activity of, I am writing VC, vascular cambium, and then types of cells formed by the activity of vascular cambium. Types of cells formed by the activity of vascular cambium. By the activity of vascular cambium. Right? Okay. Next is types of, what is the fate of, what will be the fate after secondary growth, what will be the fate of primary tissues, that is primary xylem and primary phloem present inside the stele. So fate of primary xylem and primary phloem. Primary xylem and primary phloem. And after this we will be studying about the formation of annual rings. Formation of annual rings. And after this step we will be studying about the formation of two types of wood, that is early wood and late wood. Early wood is also called as spring wood. Late wood is also called as autumn wood. So formation of two types of wood. Formation of early wood and late wood. Late wood. I am writing W, wood. Okay, next, formation of heartwood and springwood. Formation of heartwood and springwood. Sorry, sapwood. Sapwood, right? So these are different steps we are going to study about. Okay, let's start with the first step, that is formation of vascular cambium. So actually in the transverse section of dicot stem, we already studied the transverse section of dicot stem in the previous video. Go through that video, transverse section of dicot stem. So there we studied that in the transverse section of dicot stem, vascular bundles are arranged in the form of ring. So arrangement of vascular bundles in the form of ring is called as U-stele. So this is the characteristic feature. Arrangement of vascular bundles in the form of ring, that is U-stele, is the characteristic feature of dicot stem, because this type of arrangement of vascular bundles is not seen in any other organ except dicot stem. Right? So let's study how this vascular cambial ring is formed in case of dicot stem. So in case of dicot stem, what happens? These vascular bundles are arranged in the form of ring. Vascular bundles are arranged in the form of ring. Right, I am drawing vascular bundles which are arranged in the form of ring. And these vascular bundles, what is the shape of these vascular bundles? They are wedge shaped. Right? Okay. So this is the cambium present between vascular bundles. Present inside the vascular bundles, not between, present inside the vascular bundles. This is cambium. Right? Okay. Now this is phloem present inside this part is phloem. So what is this phloem? Primary phloem. Primary phloem. And now this is xylem, which is present towards center. What is this phloem? Primary xylem. What is this xylem? Sorry, primary xylem. Okay. Now primary phloem, cambium and primary xylem together is representing one vascular bundle present in dicot stem. Okay. Now, the cambium which is present between phloem and xylem of the same vascular bundle is called as intrafascicular vascular cambium, because this cambium is present inside the vascular bundle. So, as this cambium is present inside the vascular bundle, it is called as intrafascicular vascular cambium, right? So, this is intrafascicular vascular cambium. Fascicular vascular cambium. Vascular cambium, I am writing it as VC. Okay. Now, in between these vascular bundles, what type of tissue is present? Parenchymatous tissue is present. And this parenchymatous tissue which is present between the vascular bundles is in the form of rays. And these rays are called as medullary rays. Right? So, in between the vascular bundles, parenchyma is present in the form of rays. Right? So, these rays are called as medullary rays. These rays are called as medullary rays. Now, the parenchyma cells present in these medullary rays undergo de-differentiation. De-differentiation means they regain the capacity of cell division and get de-differentiated into lateral meristem, secondary meristem, that is interfascicular vascular cambium. Right? So, here I am making interfascicular vascular cambium. So, this is interfascicular vascular cambium like this, which is formed between vascular bundles. So, between all these vascular bundles, interfascicular vascular cambium is formed. It is called interfascicular vascular cambium because it is formed between vascular bundles. Hence, it is called as interfascicular vascular cambium. And what is its origin? It is secondary in origin. It is not present from the beginning of plant life. It is formed later. It is formed during secondary growth. Hence, its origin is said to be secondary. So, this is interfascicular vascular cambium, right? Interfascicular vascular cambium. Fascicular vascular cambium, and this interfascicular vascular cambium is secondary in origin, whereas intrafascicular vascular cambium, this one is primary in origin, right? I just repeat. Now we are studying about the formation of vascular cambium, which is the first step of secondary growth. This vascular cambium is formed by the joining of intrafascicular vascular cambium and interfascicular vascular cambium, right? So, intrafascicular vascular cambium is the vascular cambium which is present in plant from the beginning of plant life, and it is present inside the vascular bundles between phloem and xylem, and hence it is called as intrafascicular vascular cambium, which is primary in origin, right? And during secondary growth, medullary rays, parenchymatous cells which are present in the form of medullary rays, which are present in between the vascular bundles, undergo de-differentiation. By regaining the capacity of cell division, they undergo de-differentiation into secondary meristem, that is interfascicular vascular cambium. Why it is called interfascicular vascular cambium? Because it is formed between the vascular bundles. Hence it is called as interfascicular vascular cambium. Right? Now, after formation of interfascicular vascular cambium, which is secondary in origin, this intra and interfascicular vascular cambia join together to form a complete ring of vascular cambium. So, formation of vascular cambial ring or formation of ring of vascular cambium. So, this is the process of formation of vascular cambium. Now, let's study the next step, that is activity of vascular cambium, right?